In this video, I will show you how to include experiments into GLJ by means of the analysis of GL images. First of all, we open GLJ. We select our user. And now we can open a study or create a new study. In this case, we will open a previously created study. For instance, this one. And once that we open a study, we can notice that some icons are enabled in the GLJ interface. In particular, this experiment panel is activated. And to analyze an image, we have to press in this button that is called New Experiment. When we press that button, the first step consists in choosing an image, for instance this one, and we open the image. Now a wizard is started. This wizard consists of 4 steps: image processing, lens detection, band detection, and normalization. Each step consists of several sub-steps. As you can see, there is some help information that will guide the user in the analysis of images. The first step in image preprocessing is the cropping of the image. So to crop the image, we select the region to crop and then press the crop button. As you can see, the image is cropped. We press next. The second step in image preprocessing allows the user to invert the colors of the image, just pressing the invert button. And as you can see, the colors of the image are inverted. In the next step, we can adjust the contrast of the image both manually or using an automatic method to adjust the brightness and contrast. You can see the contrast of the image is adjusted. Finally, we can apply other operations like flipping, rotating the image, applying gamma correction, remove the background of the image, or apply different filters to remove the noise. Once that we have finished with the preprocessing of the image, we can go to the second step, that is the lane detection step. In this step, the first question that is asked to the user is whether the background of the image is dark or if it is light. In this case, the background of the image is dark. So we just select dark background. If we press next, we can see that the lanes of the image are automatically detected. In this step, we have several options. We can add lanes, delete lanes, add information about the lane, adjust the contrast of the lane, remove the background of the lane, also change colors, and then we have some options to select lanes. So imagine that this lane was not properly selected, so we can remove it. And now to add the lane, we can just select the region using the rectangle tool and then pressing the add lane button. And as you can see, the lane is added. We can change the color depending on the preferences. And for instance, imagine that we need a polygon tool to select the region, so we just press polygon and then we select the different points of the lane. To finish, we just double click and then we press add. We can also add information about the lane, so we just press a lane and we have here this button, add information, and we can add the genus, species, string number, country, and MLST information. Another option, we can adjust the contrast of an individual lane. So we have selected this lane, and if we move the contrast, we can see that the contrast is changed. The same for the background of the lane. We can remove the background of the lane individually. Once that we are happy with our selection of lanes, we press the next button. In this step, we have to fix the reference lanes of the gel limit. In this case, the reference lanes are the first and last lane. So we press first, we select the first lane and press the first fix reference lane button, and we select the last lane and press the fix reference lane button. In this step, we have to select at least 2 reference lanes. Once that we have selected our reference lanes, we press the next button. Now, we are in the band detection step, where we can automatically detect the bands of the image using a global threshold. If we move this threshold, the number of bands that are detected changes automatically. Once that we are happy with this selection, we can go to the next step. In the next step, we can fix the threshold for each lane individually. For instance, if we focus on the second lane, we can move this threshold, and as you can see, the bands that are selected are modified. And we can do the same for the rest of the lanes. Once that we have fixed the threshold for each lane, we can go to the next step. In this step, we can add bands and remove bands manually. To add a band, we just press in any region of the image and press the Add button. And as you can see, a new lane is added. If we want to remove a band, we just select the band, for instance, this band, and press the Delete button. We can also change the color of the bands depending on the preferences of the user, and as you can see, the color of the bands is changed. There is also the possibility of undoing the operations, so if we just press undo, we can see that this band that was removed previously is added again, and we have the option to redo, and then the band is removed. We can zoom the image, just pressing the zoom button and then pressing in any region of the image. Then we can move in the image with the hand button, just moving the hand. And to go back to the point tool, we just press the point, and then we can press in any band of the image. To return to zoom out, we just press zoom and then right clicking on the image with zoom out. We can also show the histogram of the different lanes, just checking this checkbox and then pressing in any region of the image. When we press in this region, the histogram that is generated corresponds to the lane of that region. As you can see, its maximum of this image corresponds to one of the maximum of the image. If we press another region, the histogram of that region is selected. If we press in any point of the histogram, an intermediary lane is selected. To add this lane, to add this band, we just press the Add button. Okay. Once that we have selected all the bands of the image, we press the next button, and we are in the normalization step. In this step, we have two options. we can select one of the available standards. 3 standards are automatically created when using GLJ, or we can create a new standard. In this case, we will use the lambda ladder marker. So we select this marker and then press the finish button. As you can see, we have an information message that says that the lambda ladder marker contains 15 bands, but the reference lane contains only 13 bands. So we have to remove the molecular weights that are not in this image. In this case, we have to remove these 2 molecular weights. And we press the OK button. And now the experiment is created. And as you can see, the experiment is added to the GLJ main panel. And if we press on this experiment, the lengths of that experiment are shown, and also the image associated with that experiment.